Hello and welcome to the latest CSF podcast on axial spondyloarthritis. We'll be bringing you new episodes on a bi-monthly basis alongside our psoriatic arthritis podcasts, and we'll also be supplying you with monthly side decks to help keep you up to date with the latest research and publications in the field of axial spondyloarthritis. First of all, allow me to introduce myself and my co-hosts. My name is Atul Deodhar. I'm a professor of medicine and medical director of rheumatology clinics in the Division of Arthritis and Rheumatic Diseases at Oregon Health and Science University in Portland, Oregon. And today I'm joined by a wealth of experts in the field of rheumatology, including Professor Hideto Kameda, professor of internal medicine at Toho University, Dr. Sofia Ramiro, a consultant rheumatologist and senior researcher at Zeidelland Medical Center and Leiden University Medical Center. And finally, Professor Xenofon Baraliakos, Professor of Internal Medicine and Rheumatology at the Ruhr University in Bochum, and a senior consultant and scientific coordinator of the Rheumatology Center in Herne, Germany. And of course, if you want to find out more about us and the papers we discussed today, please head over to the CSF website, which is www.cytokinesignaling.com. Professor Kameda.
Okay, thank you too. Our first paper discusses new and emerging treatments for SPA. In particular, it shines a light on treatments within AxSpA. Following this, our second paper sought to assess the long-term safety, tolerability, and efficacy of bimekizumab in patients with active AS. Now, over to you, Professor Baraliakos.
Yes, thank you very much both for this kind introduction, and I'm very happy to take this up. As it was mentioned, we have different papers to discuss, and our first paper is entitled Emerging Therapies for the Treatment of Spondyloarthritis, with focus on axial spondyloarthritis, and was authored by Jürgen Braun, Uda Kiltz, and myself. Now, for the study background, we need to take into account that for classification purposes, the differentiation between radiographic and non-radiographic axial spondyloarthritis is based on the degree of the structural changes that we see on the conventional radiographs of the sacroiliac joints. Neutralization of different cytokines, such as, for example, IL-17A and F, may be more efficacious than the alone neutralization of IL-17A. Development of dual and bispecific inhibitors for IL-17A and F follows this rationale, and biologic DMARDs for targeting IL-17A, they have been already approved for psoriasis, psoriatic arthritis, and axial spondyloarthritis. They include secukinumab and ixekizumab. So we sought to produce a review that aimed to discuss the emerging aspects and the emerging agents for the treatment of axial spondyloarthritis in that regard. Now, if we go over to the different cytokines that we have discussed in that review, I would like to start with IL-23 antagonists. Just for information, IL-23 itself is a heterodimeric cytokine composed of an IL-12B, IL-12P40, it's called subunit shared with IL-12, and an IL-23A, it's called IL-23P19 subunit. Inhibitors of IL-23, which have been shown to work on psoriasis and psoriatic arthritis and chronic inflammatory bowel disease and mainly ulcerative colitis, are there. However, efficacy has not been shown in axial spondyloarthritis, and also not in axial psoriatic arthritis or psoriatic arthritis with axial involvement, as we call it at the moment. For the JAK inhibitors, baricitinib has been mainly studied in rheumatoid arthritis. However, it has been successfully administered in patients suffering from atopic dermatitis, alopecia areata, and also severe COVID-19 very recently. However, it has not been studied in axial spondyloarthritis to date. In biochemical assays, filgotinib preferentially inhibits the activity of JAK1, showing a higher potency, 5-fold higher potency for JAK1 as compared to JAK2, 3, and TYK2. The preferential inhibition of JAK1 is used to justify that anemia a possible consequence of JAK2 inhibition occurs only rarely with this compound with treatment with filgotinib. Tofacitinib's mode, on the other hand, revolves around selectively and irreversibly blocking JAK1 and JAK3. We also need to know that FDA has recently released a black box warning because the results of the oral surveillance study that we, in the meantime, all know have highlighted a greater risk of malignancy in that study. Regarding now other modes of action, such as the IL-17A and F aspects that I mentioned in the beginning, bimekizumab is the drug to mention here. In a study first published in 2020, data from a population of patients suffering with ankylosing spondylitis showed a greater proportion achieving the primary outcome of ASAS40 with bimekizumab treatment over placebo. There was a significant dose response that was observed, and similar efficacy has been demonstrated for both radiographic and non-radiographic AxSpA. And further efficacy data has also highlighted a sustained improvement in disease activity over the years. This means over a follow-up of 3 years. In this investigation, nasopharyngitis and upper respiratory tract infections were the most frequent treatment emergent adverse events, as we also know them from the vast majority of biologic DMARDs. For filgotinib, filgotinib is also a JAK inhibitor. It has been shown to be efficacious and well tolerated in patients with active ankylosing spondylitis who have not responded to first-line non-steroidal antirheumatic drugs, as also the other compounds that I've mentioned. The main safety concerns of JAK inhibition therapy is cardiovascular adverse events and thrombosis, largely due to publication known here. I mentioned oral surveillance again, which have resulted in the recent FDA black box warning that I just mentioned. However, the only significant numerical differences were in serious infections, VTEs, and herpes zoster infections. And data from animal studies have raised concerns about sperm toxicity. The very recent, or the most recent data and information here is that this warning is now no longer an issue, because studies have shown that this does not really occur with filgotinib. There has been a study that was just finished, and data have been out. They will be published very soon. Now, claiming other aspects such as structural damage, modification, axial spondyloarthritis, the question is whether or not biologic DMARDs or any DMARDs are important in that indication and that topic. We know that structural damage in axial spondyloarthritis so far has been mostly considered this for the spine, since our patients have been advanced, those are the ones that have been included in studies. And there we talk about new bone formation. And several studies already have suggested that the number of erosions may also be reduced by any biologic anti-inflammatory treatment, even after a relatively short period of time. So this means that both the sacroiliac joint changes with erosions, but also the new bone formation in the spine are relevant for that topic. Now, despite a growing body of evidence, there is some disagreement on whether or not anti-TNF agents, especially, and this has been shown in more studies than in any other modes of action, may decelerate structural damage. My personal opinion is that this is indeed happening, and we do have a much lesser structural damage progression over time with those biologics, as when we compare data, historical data mainly, in patients not treated with biologic DMARDs. Now, for the conclusion overall, what we've seen in that review, and we've seen from the body of evidence that is out there, both bimekizumab and filgotinib are the ones that are represented rather emerging therapies in the field for spondyloarthritis. Bimekizumab especially is being studied in axial spondyloarthritis, psoriatic arthritis, and PSO, psoriasis. These are data that are now upcoming, and we will see very, very, very soon in our daily practice. Whilst there is an increasing body of evidence for the efficacy and safety of bimekizumab, of course, we also know that we need longer-term follow-ups to understand that drug better. And of course, for filgotinib, further studies are required to confirm the benefits. There are now attempts ongoing for such studies. Published data has shown no new safety signals for filgotinib as a JAK1 selective inhibitor. And again, sperm toxicity does not seem to be a concern based on the most recent information that we'll also by the way, affect also the safety information being given with the drug, also from the official bodies. The next set of agents in the pipeline are agents targeting IL-17A and JAK inhibitors overall, with the idea of combining agents of different modes of action needs more studies, since we know that although we have good results, also other patients, there might be patients, these patients who just do not achieve the very good response or even remission. And of course, we need more data for these kind of combinations. At the moment, this is not on the table in terms of official treatment aspects. Now, on the other hand, in contrast to inhibition of IL-17 inhibitors, JAK inhibitors are also work in RA, as I mentioned before, and there is, of course, this limitation that IL-17 inhibitors do not work in rheumatoid arthritis, even though some effect may be there. Some studies have reported some effects, but they are certainly not in the amount that we would consider them for indication. There have been overall 4 JAK inhibitors, including filgotinib in that case, that are approved for rheumatoid arthritis. And there's also several head-to-head trials ongoing with bimekizumab in plaque psoriasis. The last one showed that the bispecific inhibition that bimekizumab does with IL-17A and F may indeed be superior to inhibition of IL-17A alone. For example, when we compare it with high doses of secukinumab, this means the 300 milligrams. Whether this now may also occur in patients with access spondyloarthritis and psoriatic arthritis, we do need to understand that, and obviously study data remain to show that.
Thank you so much, Professor Baraliakos. And I was going to ask that question, what you were mentioning at the very end, as to whether the double or dual inhibition of IL-17A and IL-17F is better than IL-17A alone. And that's one of the questions that is asked by rheumatologists to me when I have presented this data also about bimekizumab in the past. And you're right that unless we do head-to-head studies, it is difficult to know whether the dual inhibition is better than single inhibition, the way it was shown in psoriasis. And we don't have those data available for either psoriatic arthritis or axial spondyloarthritis. One of the other questions I have, and that I'm going to raise and ask to Dr. Ramiro, is we are not discussing here the new ASAS-EULAR treatment guidelines, because with the expansion of these agents that we have at our disposal, I mean, there was a time when we had nothing to treat axial spondyloarthritis with apart from non-steroidals, and of course, TNF inhibitors were the big change and big bright dawn. But now we have got IL-17 inhibitor, IL-17A and F inhibitor, we have got JAK inhibitors. The question that comes up is, which one would you use first? And Dr. Ramiro, we are not discussing this here, but can you just, in short, tell us about your experience with the ASAS-EULAR treatment guidelines and how they guide us about which drug to use first?
Thank you for the question and for bringing up the ASAS-EULAR recommendations here. I think I will have to make it a very brief update on it. And actually, in patients that are eligible for treatment with biological or targeted synthetic DMARDs, actually, we are not making immediately a clear choice between them, because patients can get any of the drug classes you mentioned, so TNF inhibition, IL-17 inhibition, JAK inhibition. And unfortunately, we do not have head-to-head studies, so it's quite difficult to make a choice about which is more efficacious, comparative efficacy. But we know, and the task force wanted to emphasize that there is the current practice is to start with a TNF inhibitor and with an IL-17 inhibitor. And there are reasons for this, reasons being the more accumulated experience, more accumulated safety data, also from observational studies and from daily clinical practice. And also the fact that from JAK inhibition, we only have trial data, and with some potential safety concerns when we look at oral surveillance in population of patients with RA, although we don't know whether it really applies to patients with actual SPA who are younger, who have less comorbidities, and so on. But in short, any of the drug classes can be used, while giving preference and emphasizing that current practice is TNF inhibition or IL-17 inhibition. It's also important to look at extramusculoskeletal manifestations because they can guide us in terms of the treatment choice. Whereas in terms of actual efficacy and efficacy on peripheral disease, we do not have clear evidence of different efficacy across the drug options. In terms of extramusculoskeletal manifestations we have, and therefore we brought in a new recommendation with patients with recurrent uveitis or with active IBD. Then there is a preference towards monoclonal antibodies against TNF, whereas in patients with significant psoriasis, there is a preference for IL-17 inhibition, as these molecules have shown exactly in PSA that they have a higher effect on the skin.
Yeah.
And I think I would like to keep it here to not extend too much into the recommendations.
No, thank you.
Thank you.
That's wonderful. Well, why don't you continue, Dr. Ramiro, with our second paper for this podcast?
Yes, sure. Thank you very much. So over to our second paper entitled Safety and Efficacy of bimekizumab in Patients with Active Ankylosing Spondylitis, 3-year results from a phase 2b randomized control trial and its open label extension study, which is authored as the first author, our colleague Xenofon Baraliakos, and several of us in this podcast have also contributed to co-author it. So we have just heard an introduction on bimekizumab, so I will not repeat. We are aware of it being an IL-17 inhibitor with dual inhibition of IL-17A and F, and that clinical studies have shown efficacy in psoriasis, even with showing superiority against multiple therapies, also efficacy in psoriatic arthritis and in actual SPA. And we have here still the phase 2 study that was conducted several years ago, so the BAGILE study, and now in which it was demonstrated that there was a rapid and significant reduction in disease activity at week 12 with bimekizumab in patients with active ankylosing spondylitis. And now in this study, we are looking at the long-term safety, tolerability, and efficacy of bimekizumab up to 3 years of follow-up. So patients with active radiographic axial SPA, or also formerly known as ankylosing spondylitis, who completed the dose ranging 48 weeks BAGILE randomized control trial were eligible to participate in this open label extension, in which participants received 160 milligrams of bimekizumab every 4 weeks. And here we are looking at safety and efficacy results through 156 weeks, so roughly 3 years. Missing data were imputed using the most conservative approach, so non-responder imputation for binary outcomes and multiple imputation for continuous outcomes. And so now moving to the results. From baseline until week 156, 280 of the 303 patients experienced at least one treatment emergent adverse event. This brings us into an incidence rate of 141 per 100 patient years. The most frequent adverse event, as common as we see in these biological therapies, was nasopharyngitis with an incidence of 8.1 per 100 patient years, and upper respiratory tract infections with an incidence of 5.0 per 100 patient years. Additionally, 67 of the 303 patients had mild to moderate localized fungal infections, so giving an incidence of 9.8 per 100 patient years. And this fungal infections so were mainly Candida infections, 3.7 per 100 patient years, and 23 of the 303 patients had oral candidiasis, so 3.0 per 100 patient years, while 10 patients had serious infections, 1.3 per 100 patient years, and no cases of active tuberculosis were reported. As we are used to seeing that extramusculoskeletal manifestations are reported as safety signals in trials, so this was still the case in this trial. This will possibly and hopefully change in future trials, but for now we have here the information on active inflammatory bowel disease with 1.1 per 100 patient years, anterior uveitis 0.7 per 100 patient years, and MACE, so major adverse cardiovascular events, which were adjudicated, 0.3 per 100 patient years. So these were all infrequent. About efficacy, we can say that it was demonstrated until week 48, and then it was sustained in the open label extension. If we look at week 156, so 3 years, the non-responder imputation analysis showed 54% of patients meeting ASAS 40 criteria. So that's 73% when we look at observed cases. And 28% of patients achieved partial remission. That's 38% of observed cases. ASDAS reduced from baseline with a mean of 3.9 to week 48 with a mean of 2.1 ASDAS, and week 156, we had a mean ASDAS of 1.9. Patients showed sustained improvements in pain, fatigue, physical function, and health-related quality of life. So in conclusion, the safety profile of bimekizumab was found to be consistent with previously demonstrated findings, and no new safety signals were identified. Bimekizumab delivered sustained long-term efficacy in patients with AS, including reduced disease activity, improved patient function, and improved quality of life. And overall, these results support bimekizumab as a potential treatment option in axial spondyloarthritis. And as we have just heard, there will be news soon in terms of phase 3 trials. So stay tuned. We will have updates on that soon as well.
Okay, thank you, Sofia, for your very nice talk. And because Atul already raised the comparison between AxSpA inhibitor versus bimekizumab, I want to raise another question. So bimekizumab versus brodalumab, it's a AxSpA receptor A inhibitor, so it actually inhibits AxSpA F and C. So what do you think about the comparison between bimekizumab versus brodalumab?
Thank you very much. That's also a very good question. I have to tell you that what we know is at this moment only data from laboratory and from how these cytokines behave, but we do not have any idea in terms of comparison in humans. So until I see any comparative data, I cannot pronounce about different efficacy of these molecules. And I would love to know whether there is superior efficacy of one versus the other, but as I don't know from bimekizumab compared to secukinumab or izokizumab, I don't know compared to brodalumab either. And I sincerely hope that that will be then the future and the next phases that we will see in actual SPA, that we start seeing more head-to-head trials that can truly inform us in terms of the treatment algorithm and inform updates of future recommendations.
Thank you for joining us for this AxSpA podcast brought to you by the CSF. We really hope you found it useful. If you did, don't forget to subscribe to our channels on YouTube, SoundCloud, Spotify, or wherever you get your podcasts from, so that you don't miss any future episodes. If you want to read more about what we have discussed today, head over to cytokinesignaling.com, where you'll find detailed summary slides of each of the papers. See you next time.